Hello, everybody, and welcome to today's episode of High School of Mathematical Oncology, where we explore how science, mathematics, and medicine are coming together to create the future of cancer treatment. I'm thrilled to be joined today by Dr. Alexander Anderson, Chair of Integrated Mathematical Oncology at Moffitt Cancer Center, and one of the pioneers of adaptive therapy and mathematical oncology. Dr. Anderson's work is transforming how we treat cancer, not just by studying biology, but by using mathematical models to predict, personalize, and even outmaneuver this disease. Dr. Anderson, thank you so much for being here.
Great to be
So
here.
first question, let's start with your journey. A lot of students think about math and science, especially cancer treatment, as sort of different things. What inspired you to use mathematics to fight cancer, and how did you find your way into this field?
That's a great question. So when I was younger, I was sort of impressed by the idea that mathematics could sort of describe and help us understand biology. And that sort of drew me towards doing courses that were linking mathematics and biology. And probably the first sort of taste that I got of that was through looking at fractals and understanding chaos. Both of those sort of dynamical systems, the idea that you could get very complex structures out of very simple equations is something that was kind of impressive to me, mind-blowing even. And, when I was doing my undergrad for my honors project, I focused on fractals and trying to understand if there was patterns in the structure and this whole concept of self-similarity. And that then ultimately led me down the route of dynamical systems towards chaos. And that's where I found a master's in mathematical biology at Dundee University. And that was where I started to hear more about mathematics being applied to biology, including medical applications. So modeling tumor growth, modeling sort of neuronal firing, modeling population dynamics, and the kind of complex interactions that very simple equations can capture. And so from there, I then did a PhD in mathematical biology, and that was actually on nematode movement. And nematodes are small worms, which are a real pest in crops in the UK, particularly potatoes. And during that time, I developed this model, this technique, which was basically, I called it the hybrid discrete continuum technique, which essentially linked discrete agents with continuous chemical fields. So it was like a PDE meets an agent-based model. And that merging, that tool that I kind of developed then ultimately is something that we've used throughout my cancer work. So I didn't actually start working on cancer until I moved to my postdoc. And that was my first postdoc was with Mark Chaplin at Bath University. Well, he was at Bath at the time, and I worked on angiogenesis. But as soon as I started working on cancer, I realized there was so many interesting problems there and opportunities for mathematics and for mathematical modeling, and really caught my attention. So that's kind of the trajectory that took me there.
So, like going to the know of like those cancer models, you used analogies comparing them to predicting a hurricane or like an evolving ecosystem to understand how to treat cancer in this way. How do these metaphors help both scientists and the public better understand what makes cancer just so hard to treat?
Yeah, I mean that's a another great question. I think these are two different metaphors, but the whole idea of the kind of weather prediction and like, we often talk about the hurricane cone of uncertainty. So, in Florida, hurricane season is just beginning. One of the things that you see often in the weather is where the current eye of the hurricane is, and then this cone that emerges from it. And of course, the cone is actually calculated from many mathematical model simulations that show where that potential eye might go next. And the reality is it's uncertain, and that there's many factors that can impact the direction of that hurricane that are both to do with the fluid and the land and the air that the hurricane's moving through, but also through some stochastic interactions that might occur within the hurricane itself. And so this idea that you might know exactly what the current state is and you can have some idea of where it might go in the future, but there's uncertainty, that's very relevant to cancer. So, for example, when we're trying to understand the best way to treat a patient, there's no single best way because we can't be certain about every aspect of the patient's disease. So we have to capture some aspect of it and then account for the fact there's this uncertainty that exists. And so if you can come up with a treatment strategy that will take account of all of that uncertainty and still work, then that would be a very good strategy to take. Okay. But sometimes the uncertainty is too large, so you have to wait for more information. And one of the great things that you can see when they're predicting these hurricanes, the more points that they get on that trajectory, the narrower the cone of uncertainty becomes, the better we become at predicting the future. And that's very true of a patient. The more data, temporal and spatial data that we have on a patient's disease, the better we become at predicting the future of their disease. And then the ecological aspect is kind of important because historically cancer was viewed purely as being a collection of mutated cells. And, you know, in some ways, it's probably easy to imagine cancer as this alien thing that is just a ball of these mutated cells. But in reality, those cancers are often within fully functional organs that, and it's slowly damaging and eating away at one of the part of those organs or one of those organs. So, you know, people with lung cancer can have a lung that's heavily infiltrated by cancer, but they're still breathing. And people with brain cancer can have a large tumor in their brain, but still be functional. Now, at some point, those functions start to deteriorate, but the point is it's the interaction between the tumor and the non-tumor that is also important for that disease. And of course, as we've come to realize, the immune system is part of that normal, and it's something that we're trying to exploit now. But of course, the tumor isn't just interacting with the immune system, it's also interacting with the other cellular and microenvironmental components like fibroblasts or blood vessels or you know, other cells and say factors, growth factors and nutrients that are in the environment. And so we need to look at the ecological aspects as this collective interaction between the tumor and the non-tumor. And as we've done that, we've started to understand more and more about what separates tumors that are bad and hard to treat and tumors that are, well, all tumors are bad, but tumors that are easier to treat. And for example, one of the things that we're able to do now is take a pre-treatment biopsy, and based on the kind of spatial orientation and the numbers of immune and tumor cells and how they're sort of interacting, we can predict whether they'll respond to immunotherapy or not. And so it's already kind of hard coded in that spatial pattern and the interactions, whether they're going to respond or not. And so that's something that we could never have done without mathematical models and without the data to feed those models.
Yeah. That's actually I'd like to go back to the whole tumor versus non-tumor analogy, or the tumor versus non-tumor idea. In your work you've used adaptive therapy, the idea that sometimes you don't want to like maximize the dose because then you kill all the
Yeah.
susceptible tumor, but that leaves the resistant tumor to grow and spread, and eventually you can't do much about it anymore. So why might giving less drug be more effective than maximum doses in cancer therapy?
Yeah, I mean, I think you just said it, which is basically if you give the maximum tolerated dose, and in a scenario, if we imagine we could kill every cell that was sensitive to that drug, all you've left behind is all the resistant cells. And if they're resistant, that means it doesn't matter how much drug you give now, they're resistant, they just grow. And so this stems from a kind of concept that Bob Gattenby developed here at Moffitt. He's a radiologist, but has been heavily involved in mathematical modeling. And basically, his idea was if we, instead of giving maximum tolerated dose and eradicating everything, we leave behind some of those sensitive cells to compete with the resistant cells. Now, it does mean that you're not going to eradicate the disease. You're not going to cure the patient. But, all of the sort of cancers where we're doing this are cancers that are not curable, that we know are going to fail because they're already metastatic and they're not curable. So how about instead of trying to cure the incurable, we try to control the incurable? And so adaptive therapy is really a control strategy. And the idea is to use the sensitive cells to compete with their resistant counterparts to suppress that resistant population more effectively. Now, the resistant cells will always continue to grow. There's nothing you can do about that. But if you can at least have the resources that they're utilizing be competed for by the sensitive cells, then you can potentially let that tumor grow and shrink it with drug and then let it regrow and shrink it with drug. And to do that, you have to switch the drug on and off or increase the dose and decrease the dose, right? And so the whole idea of adapting the therapy, you're adapting it to the individual dynamics of the tumor. So you're either de-escalating or escalating that dose as you see the tumor grow and shrink. And it could be that you're thinking about this from in terms of switching the drug on and off, or you're thinking about this from reducing the dose or increasing the dose. Both can work. And we've done this across a number of different cancers. Now, another advantage of that potentially is that the tumors getting less therapy, but that also means the non-tumor is getting less therapy. So the patient actually feels better within themselves because they're having less drug. And some of these drugs that we're treating the tumors with are quite toxic. So the whole idea of reducing the toxicity, increasing the quality of life is another added benefit of something like adaptive therapy. Now, it doesn't come without problems because one of the problems potentially is that you're carrying a larger disease burden for longer. So if you're trying to have your sensitive cells compete with the resistant cells, that means there's more tumor in your system. And for some patients, that might be unacceptable. It might be too painful. It might make them more sick. But if we can extend their life and improve the quality of life for the bulk of patients, then it's a worthwhile strategy. And so we've done this now in about 6 different cancers. We have 9 active clinical trials where we're using this, everything from skin cancer to prostate to lung to breast, um, a whole range of different disease settings. And within those subsets of cancers, so for example, in skin, we're also looking at melanoma and basal cell carcinoma. One of the things you need to make this approach work is a way to measure the disease, right? To measure it ideally in a non-invasive and easy to do way. So for example, from a blood sample. So in prostate cancer, you can get a measure of what's called PSA. In ovarian cancer, you can get a measure of CA-125. But for things like lung cancer and breast cancer, there isn't necessarily an easy biomarker that you can measure in the bloodstream that'll give you an idea of how much tumor is present. And so then you've got to rely on other sources like imaging, clinical imaging of the whole body, or maybe something like ctDNA, which is a kind of newer emerging technology, which essentially looks at fragmented DNA from the tumor in the bloodstream. But it's sometimes difficult to interpret precisely what that means.
Yeah, and I guess like knowing precisely what it means would be important because of what you said. Like, there's a Goldilocks window in like trying to get enough treatment to keep the tumor down, but not enough so that it kills all the sensitive cells. Could you explain how tumor metabolism plays a role into both immune evasion and treatment effectiveness, and sort of how that plays into adaptive therapy.
Yeah. Okay, that's a lot to unpack there. But one thing we know about most cancers is that they're glycolytic. So they use Warburg, and essentially that means that they utilize both glucose and oxygen as their nutrient sources. Whereas most of your normal cells in your body use a tiny amount of glucose and largely rely on oxygen, right? And so cancers have sort of hijacked this pathway to gaining more energy. Um, but the kind of byproduct of that sort of more glycolytic behavior is that they produce acid. So it's lactic acid is a byproduct of that glycolytic metabolism. And that acidity, ironically, is bad for the normal tissue. So um, tumor cells that are highly glycolytic are also therefore quite acidic, which means the environment around the tumor is quite acidic. And that can hurt normal epithelium, normal cells. Your normal tissue doesn't like it, can kill those cells. But the other problem is that immune cells are effectively silenced in high, in a sort of acidic environments. And so what people probably don't realize is that your lymph nodes are actually quite acidic. And we believe part of the reason might be that's because we don't want your immune cells attacking them, their home, right? Their lymphatic system. And so that kind of control that pH has on the immune cells is effectively being hijacked by the cancer indirectly when it's becoming more acidic because it's using this glycolytic pathway. And so we've hypothesized an idea of potentially giving some sort of therapy that could buffer away that acidity, reduce that acidity, making it, let's say, more of a nicer environment for the immune cells and a harder environment for the tumor. And so a simple way potentially to do that is to use a buffer like sodium bicarb or a drink, one of these sports drinks that sort of helps neutralize the acidity in your system. And Bob has done some preclinical experiments where we fed drinking water in the drinking water of animals and showed that just by putting bicarb in the sodium bicarb in the animals drinking water, they don't get cancer, even though these are animals that would spontaneously grow tumors. And so it's a little bit sci-fi to think this is going to be a clinical trial, but Bob actually did start a clinical trial just before I came to Moffitt in 2008 that was trying to use sodium bicarb as a therapeutic strategy. The problem is you had to drink gallons of it. But now with modern technologies and there's new ways to get quite high levels of these sort of let's say pH neutralizers into your system much easier. And so that could be a viable strategy for the future. So one could imagine a therapy where you might give a drug that targets the tumor, and then you give another drug which in fact targets the environment of the tumor that neutralizes that acidity that helps facilitate an immune attack. And then you could potentially switch between those two in a way that would be exploiting the kind of adaptive approach where you would be targeting the tumor on one hand and then, like facilitating regrowth of the sensitive population on the other, because often the more resistant cells are the more glycolytic cells. So given that buffer could help facilitate that process. So that would lead then to kind of adaptive therapy where you're targeting the context in one hand or the environment and targeting the tumor in the other and playing between those two to maximize your control of the disease.
Yeah. So going off of like the idea of switching between drugs, I feel like for a lot of people it would feel like the intuitive thing is to just like use both drugs at once, try to maximize the treatment and boosting the immune system. But like what if the mathematical model says that's not the best play? Like how do you approach these conversations with doctors or patients? Like a strategy that the math said should work, but doesn't seem as intuitive to people.
Yeah, so I think that it's cancer isn't intuitive and it's not linear. And that's something that's hard for the public to sort of grasp in some sense, that the idea that if I give more drug, I'll kill more tumor. Um, and but if you can sort of at least accept that maybe there's cells that aren't sensitive to that drug. But if we come back to the concept that if I have two treatments and one of them's immunotherapy, um, the timing of when you give that immunotherapy should be more carefully thought out. And part of the problem is that historically, drugs, when they're given in combination, are given simultaneously at the same time. They're not given in any sort of strategic manner, where, for example, one could imagine if I give drug A first, then drug B, that could potentially give me some benefit, because if drug A deliberately targets a population that leaves behind cells that are now more susceptible to drug B, then that order would be important. And this is what we've sort of hypothesized with immunotherapy, that giving immunotherapy up front before you give that targeted therapy could actually be a better strategy than giving it simultaneously, right? There's still, you can have it overlap, but the point there is you're priming an immune response. You're already waking up the immune system before you go in and start hitting the tumor and killing a bunch of cells. And in fact, as a result of that sort of priming, you now get a reaction to that material that's been destroyed by the treatment. That dead cell material could now educate that immune population you've just woke up to then go after the remaining cancer. So I think the order and sequence of drugs is something that needs to be better thought about and understood. And historically has only been done together because that's what clinicians traditionally have done. Now, when it comes to trying to explain this to patients, I think one has to just show them the facts and explain that as clearly as you can. A good example of this was in the prostate cancer trial, where we were doing adaptive therapy with this drug abiraterone, and the standard strategy is to give that at maximum tolerated dose until essentially the patients fail the treatment, and that's often within a year on that therapy. Um, but the adaptive strategy, and this was largely driven again by Bob and a clinician here, Jinsong Zhang, and Joe Brown was also involved in that, and essentially came up with a very simple rule of thumb, which is, we called it AT50, which is that you reduce the tumor by 50% of its size, then you stop the treatment, and then let it return to its original size, and then start the treatment again. And so an obvious question there is, why did you use 50%? Why not use 60% or 30% or, whatever, a different number? And there was a practical reason, which was all the models actually showed that if you reduce the tumor the minimum amount, so like say 10% of the size, and then let it regrow, that would be the best way to control it. Essentially keep it almost at maximum density the entire time, its maximum capacity. But of course, that's not practical, and patients also don't want to hear that. They don't want to say, well, your tumor's responding, we're going to stop treatment now. But if you said your tumor's responding, we're going to stop it once it shrinks by 50%, that feels and sounds a bit more doable, right? And so again, that comes to this issue of how do you convince the public that this strategy makes sense, right? And so yeah, there has to be some compromise, even if that isn't the optimal strategy, that isn't the best one. It's still better than giving maximum tolerated dose.
Yeah, in your research studies, you found that when mice receive genetically identical cancer cells, they can still have dramatically different responses. So, what causes this kind of variability, and how can mathematical models make sense of it?
Yeah, that's a very kind of on-topic question right now. Um, are animals, for example, the best experimental system to be looking at therapies and to be looking at tumor growth dynamics that are relevant to a patient? And so one of the issues that we have is that depending on the approach, and I think that experiment you're specifically talking about was done by one of my colleagues here who injected melanoma cells into the mammary fat pad of an animal. And so first of all, you're injecting human cells into an animal, and you're putting those cells into a part of the animal where cells don't normally exist, right? And so you're injecting the same number of cells. These cells are supposedly genetically the same. But of course, what we've learned over the years is that even cell lines are heterogeneous. And what I mean by that is even if genetically they have a similar readout, and of course people have done some single cell analysis of cell lines, and they've found that there's distinct epigenetic profiles present within there, but somehow they're coexisting, right? There's no single winner, at least in the dish. But you take a cell line and you put it inside an animal, it's a very large selection pressure where whoever gets that kind of edge, whoever happens to be better proliferating or maybe surviving in this very difficult and distinct environment from which they're from, then that's the population that's going to win. And depending on where exactly that's put and how many cells are in there and how the animal reacts to those cells, you can get very different dynamics that emerge. And that's a kind of accepted thing when working with animals, which is the animal tumors can grow quite differently, right, without any treatment. And so that's something I think and then how they respond to treatment can also be quite distinct. So there's been a move, I think, over the last few years that's really coming, I think, to a head this year of sort of embracing what we're calling non-animal models, right? So that of course includes mathematical models, but it can include patient-derived organoids or patient-derived samples where you can keep a piece of a human tissue alive for a period of time in the lab. It's moving away from animals and moving more to in silico and sort of, let's say, ex vivo type approaches. So, you know, there's an understanding, I think, now that animals can potentially give us the wrong signal, give us the wrong information. They're still a useful tool, but we have to be careful of how we interpret them. And, many drugs have shown amazing success in animals, cured animals, but had then had no impact on patients, right? So that's another indicator as to why maybe these models, because animals are models of human disease, aren't as good as what we used to think they were.
I guess could it possibly due to like the spatial shape of a tumor, like your carpet patch hypothesis deals with how shape might affect a tumor?
Yeah, I mean, I think that how well mixed different subpopulations are within the tumor, if they're clustered together in patches or if they're well mixed, that could create very different dynamics. And so, years ago, we published this paper, a very simple idea where if you just take, say, 100 cells and you seed them randomly in a domain, versus if you place 100 cells together in a cluster in the middle of the domain, they will grow very differently. And the reason they'll grow very differently is because those 100 cells in the middle are immediately competing for space with each other, whereas the 100 cells spread out have very little competition, so they grow exponentially. But eventually they'll start to have competition. And so, that very simple analogy, I think, shows how spatial interactions can impact growth, but they can also impact migration and, nutrient availability and other aspects of the disease.
Yeah, I guess there's a new software, Visual PDE. It's been really good at helping me understand like how space sort of affects the tumors. I'd like to talk about your work on the evolutionary tumor board. How does that work and how is it different from a traditional cancer care team?
Yeah, so, one of the, let's say, frustrating aspects of working on cancer is that for any change in the clinic to occur, the only way to do that is to drive a clinical trial. And so, I talked about we have these 9 active clinical trials. Some of them have been ongoing for 7 years. Some are in year 5, some are in year 4. It takes years for a clinical trial to start and finish. And so you're waiting a very, very long time to get the result. And then that result might then drive a bigger clinical trial that eventually could lead to maybe significant clinical change in what we call the standard of care. But it's a long and laborious process. Whereas tumor boards are something that happens across all diseases, and they're typically for basically a mixed discipline of um, oncologists, pathologists, radiologists, and any other people involved in the care team for a given patient who has a difficult disease, a difficult cancer in that setting. So say that it was the breast tumor board, and they would get the team together to discuss that patient's specific disease and come up with a strategy, a group think on how best to deal with that patient because they don't fit maybe with standard of care or they're different for some reason. And so our idea was, can we put mathematicians and mathematical oncologists in that care team on the tumor board, along with evolutionary biologists, to then come up with a mathematical model that could forecast, predict what would be the best strategy for that patient. It's now a much faster thing, right? You basically hear about the patient the week before, and then you have to come up with a plan and a strategy within a week. So it's not years in the making, it's a week. And so that tumor board, one of the hardest things to get that up and running, and this is work that really was led by Mark Robertson-Tessie in my lab, and now he's sort of independent faculty here at IMO. Essentially, the hardest thing there was to get the clinical data in a way that we could actually see the temporal dynamics of the disease. So it sounds like an obvious and easy thing to do, right? The patient's coming in for treatment. What was their previous treatment and their treatment responses? Can we see that, please? Right? And it turns out, one, it wasn't easy, and two, the clinicians don't typically have that information at their fingertips while they're about to make that decision. So one of the things that took us the most time was to actually generate a visual plot of all of the patient's diseases, all of their metastasis over time, how the burden of those metastases changed, and what the specific treatments they received over the course of their therapy. As soon as we created that graphic, that was immediately a kind of revelation for the clinician because they could instantly see what the patient was doing. But for us, it was critical for us to be able to build any kind of model, right? We have to see the dynamics of the patient's disease. And so that, you know, visualization was central. But once we had that data, you can't just build a model and make a prediction on a single patient's trajectory, right? You need other data. And so depending on the specific cancer and the specific types of treatment, we then would go back and look at historic data for patients at Moffitt who'd got those treatments and had similar diseases. And then we'd have a cohort of patients. And so effectively what we would do is we would match the individual patient dynamics from historic data to a mathematical model, almost creating what's been termed a digital twin. So we would match that we'd map our real patient dynamics to a mathematical model. And then we'd take those individual patients and create a virtual cohort of patients that would then help forecast the real patient that we're trying to treat, right? And so that's essentially what we ended up doing. And now we've sort of done about 25 patients across a number of different disease sites. Obviously, each disease site takes a lot of work because you have to gather all of that historic data and measure all the tumors and all their lesions and get as much data as you can. And depending on the disease site, that could mean you're also building new models and new approaches. So it can be a lot. But once you've done that once, once you've kind of tackled that disease site once, you've potentially got a lot of historic data and a lot of, like, say, pre-existing tools now that you can apply when you get new patients that come into that setting.
Yeah. Like the last few minutes of the Zoom, I'd like to ask one quick question about the high school internship program at Moffitt.
Yes.
What inspired you to start this program, and what do you hope students take away from it?
Well, truthfully, it was Heiko Enderling, who used to be an IMO, and what hadn't realized is that one of my PhD students actually had previously been a high school intern at a program that Heiko was involved in when he was in Boston. So Derek Park was one of my PhD students that ended up going to Oxford, and he had done a high school internship with Heiko back in these Boston days, and that inspired him in the future to think about coming to do a PhD in mathematical oncology. So Heiko basically we thought it would be a good idea, and I thought it was crazy on some level, but agreed on another, that because we were seeing more and more sort of, let's say, junior researchers, like students that were coming out who really hadn't been exposed to the idea that mathematics and oncology could work together, right? Could be something that could be a future career, was to go and catch them at the school level where they're still thinking about what their focus should be, what their major at university should be, and encouraging them at that point to say, look, you can take a double major in maths and biology, or you could think about doing medicine, but also bring in mathematics. Don't think that you can't do both of these subjects together. And so that, historically was something that most people that major in the life sciences tend to avoid mathematics. If you're going into medicine, if you're going into biology, you're avoiding mathematics. And so that was really part of the motivation, was to change that mindset, but also at the same time give really kind of maths geeks who are really into mathematics and thinking about a career in that an option to really show how they can use that tool set in cancer and specifically in cancer research. And so the program essentially brought the best high schoolers in from across the country. We started doing it locally, but we got so many calls from people outside of Florida that we made it kind of all across the US. And bringing them and assigning them to an individual lab to spend 8 weeks doing a small research project. And it's not like just kind of boring. They actually do real science, and it's a proper research project. Now, it's a lot of work for that student, that high school student, but it's also a lot of work for their mentor trying to guide them through that process, because some of these students might never have thought about biology this way, or they might never have coded in that specific language. And so a lot of what we did at the beginning was develop.
I guess welcome back. It just cut off there, but please.
Yeah, so basically I was just saying that, the high schoolers do actual work in the labs. It's a research project. And we've even had multiple publications with high schoolers emerge from this. as a result. And so it's an intense program, but it's kind of been hugely successful. And with Heiko moving then to MD Anderson, Renee Brady has taken that over, and it continues to be hugely successful. We got something like 200 applications for essentially 15 spots. It's very competitive, and people are excited in it because, of course, it gives you almost like a research opening before you even step into university. And many of the students have went on to very nice schools, in part because of their brilliance, but also because they've already experienced this sort of research environment that they want to embrace further in their career.
I guess one last thing before we wrap up this meeting. What do you hope to see in the next 5 years of mathematical oncology? Just
So,
in,
what I'd love to see is the IMO, my department, isn't the only department doing mathematical oncology. There is some key people across the US and across the world, and we've made a big effort, I'm sure you're aware, of creating resources for mathematical oncology in general. There's a website, mathematical-oncology.org, that has all the information. We run a weekly newsletter. Well, Jeffrey West does with a whole team from different places, and we have this cool cover art that's done, and Maxi Strobel's been sort of largely leading that charge. And then we have, videos or YouTube videos on lectures. And now we're about to run a conference on mathematical oncology at the Don Cesar in St. Pete. And of course, we run our annual workshop. So the way I see it is we're trying our best to get mathematical oncology out there and get people involved. And so what I'd love to see is that there's more departments dedicated to this, and importantly, that every cancer center in the US has at least one mathematical oncologist on the payroll. Many of the top 10 cancer centers already do, and there's already some, prominent scientists at those places, but they often tend to be just a single PI that's doing that type of science, not a whole department like we have in IMO. And so I think there's enough out there that there could easily be a maths department in every cancer center, and that would be something that I think would be amazing, both for cancer research and for the mathematical oncology community. But I think it's really the future of what we need if we want to make progress in how better to control cancer.
so, Dr. Anderson, thank you so much for sharing your insights today. It's been really interesting hearing how math and medicine can come together to tackle cancer. For everyone watching, if you're curious about research math biology or how science can change lives, I encourage you to explore Dr. Anderson's lab or apply to Moffitt's high school mathematical oncology program. Links will be in the description. Thank you all for watching. I hope this sparked your curiosity and inspired you to explore more about mathematical oncology and cancer research.